Prokaryotic cells, kingdom Monera, and are commonly called as monerans. Bacteria is the most common example of prokaryotes. Bacteria are found in hot springs, icebergs, soil, intestine, seawater. It is ubiquitous in nature. Each and every place we find bacteria. Other examples of prokaryotes are Mycoplasma, PPLO, pleuropneumonia-like organisms, cyanobacteria, blue-green algae, spirochetes, and rickettsia. Sir Anton von Leeuwenhoek, he is the first to observe or discover bacteria in pond and tartar of teeth. Size of prokaryotes. Dialyster, it is the smallest bacterium. It is 0.15 to 0.3 micrometer in diameter. Spirochete is the largest bacterium, 500 micrometer in size. Bacillus, its size relies from 0.3 to 15 micrometer. Sir E. J. Cohn described 4 shapes of bacteria: Coccus, Bacillus, Spirillum, and Vibrio. Coccus is spherical bacteria. It is Monococcus, Diplococcus, Tetracoccus. These are the shapes of the bacteria, and one such shape is coccus shape. Now Bacillus, it can be rod shaped. It is rod shaped. Monobacillus, this is Diplobacillus. Spirillum, it is spiral shaped, and Vibrio, it is comma shaped. Now the general structure of a bacterial cell or a general structure of a prokaryotic cell. Let's consider this as bacteria. So the outermost covering is the cell envelope, and it is formed of 3 layers. So cell envelope is having 3 layers. One I have mentioned with yellow color. This is another layer, and finally the innermost layer is this. So these 3 layers together forms the cell envelope. So the outermost covering, known as the cell envelope, actually this is the outermost covering, and it provides shape and rigidity to the bacteria. It is formed of 3 basic layers, as I mentioned earlier, specialized to perform specific functions. The yellow color is the outermost layer of cell envelope, known as the glycocalyx. It is the outermost and differs in chemical compositions and thickness. In E. coli is a bacteria, gram-negative bacteria, Escherichia coli. This glycocalyx forms the loose mucilaginous coverings, slimy layer, and it protects the bacterium from chemical desiccation, that is attack of any foreign chemicals, and phagocytic action, and also helps in adhesion. So this layer of a cell envelope, which is the glycocalyx, have so many functions like protection, adhesion, and it forms the outer layer of the cell envelope. So chemical desiccation, as I mentioned, is protection from the foreign chemicals, and phagocytic action means if a bacteria enters in a body known as the host, so the host defense mechanism will try to engulf the bacteria, and this layer glycocalyx will protect the bacteria from this phagocytic action. So, and when this glycocalyx becomes hard and tough, then it becomes capsule and thereby it resists phagocytosis. So 3 layers: outside is the glycocalyx, then cell wall, and finally the innermost layer is the cell plasma membrane, which I will discuss in the later slides. Now the cell wall. If this is the bacteria, the outer slimy layer is the glycocalyx. Now next to glycocalyx or inner to glycocalyx, this is the cell wall. It is inner to glycocalyx and it is protective, rigid and supportive in function because it is a part of cell envelope, so its definite function is protection, rigidity and supportive in nature. And it is formed of murin and or peptidoglycan. You should remember that this cell wall is formed of murin and peptidoglycan. Now what is peptidoglycan? It is formed of polysaccharides like acetyl glucosamine and acetyl muramic acids. So this cell wall is actually, let's just take this, consider this as a cell wall. So it is formed of polysaccharides of acetyl glucosamine and acetyl muramic acid. Okay, so based on the cell wall, bacteria are further classified as gram positive and gram negative. So up to this we have discussed that this cell envelope is made up of 3 layers, the outermost being the glycocalyx, next is cell wall which is formed of peptidoglycan, and this peptidoglycan, if this is the peptidoglycan, and this is formed of polysaccharides like acetyl glucosamine and acetyl muramic acid, and with this cell wall, bacteria can further be categorized into 2 types, gram positive and gram negative. So difference between gram positive and gram negative bacteria: gram positive absorb gram stain and appear purple colored, and gram negative do not absorb gram stain. Mesosomes is present in gram positive, mesosomes absent in gram negative. Cell wall in gram positive is monolayered and cell wall in gram negative is double layered. 100 to 200 Armstrong thick cell wall is present in gram positive and 70 to 122 Armstrong of cell wall is present in gram negative. The most common example of gram positive bacteria is Diplococcus pneumoniae and gram negative is E. coli, Escherichia coli. So what is gram stain? Actually, gram stain, gram positive, gram negative, this I will discuss elaborately in another playlist known as microbiology. But with this presentation, just you remember the difference. This gram stain is actually a staining procedure to color bacteria, because if you don't stain the bacteria, it is not visible under the microscope. So to make it visible under the microscope, staining is important, and this staining is named as gram stain before the name of the scientist, that is Hans Christian Gram. So in gram positive bacteria absorbs crystal violet, so it is mentioned it absorbs gram stain and it appears purple in color under the microscope, but gram negative bacteria do not absorb gram stain. Mesosomes are present in gram positive bacteria and mesosomes are absent in gram negative bacteria. These mesosomes I will discuss in the next slide. Cell wall of gram positive bacteria is monolayered and gram negative is double layered. Just a brief introduction. This purple colored bacteria is gram positive because it has absorbed gram stain, and this red color is gram negative. Example, E. coli. Just you remember this up to for this lesson. Okay. Now the last layer of cell envelope, which is the plasma membrane. Let me discuss. If this is the bacteria, so if this is the cell, as I mentioned, the outside covering of the cell is the cell envelope. So this is the glycocalyx. Inner to glycocalyx, we have the bacteria have the cell wall, and inside to cell wall we find plasma membrane. This plasma membrane is the innermost layer, and it is thin, transparent and semi-permeable in nature. Semi-permeable means it is selectively permeable to specific substances in and out of the cell. So it is present just outside the cytoplasm. So if this is the cytoplasm, which is a jelly-like substance present inside the cell where cell organelles are embedded, and next to just outer to this cytoplasm we find the plasma membrane. Plasma membrane is found just outside to cytoplasm. It is trilaminar, means it has 3 layers. It is made up of 3 layers, and plasma membrane is made up of lipid and protein, so it is lipoproteinous. Now what is lipid I will explain in another playlist, which is biochemistry. Now this plasma membrane also consists of some enzymes that helps in bacterial respiration. Now the function of plasma membrane, it is selectively permeable to specific materials, so it exchange of specific materials is the main function of plasma membrane. Plasma membrane also has receptor molecules that helps the bacteria to detect and respond to chemicals. This plasma membrane have some receptor molecules which detects and respond to some chemicals present outside the cell. Mesosomes and chromatophores. Mesosomes, these are formed by plasma membrane folding, only happens in gram-positive bacteria, as I have already discussed in the slide where I have discussed difference between gram-positive and gram-negative bacteria. So mesosomes helps in DNA replication and their separation in daughter cells. Mesosomes increases the surface area for respiration. It helps in cell wall formation. So if this is the bacteria, inside we have cytoplasm, right? Okay. Now outside there is a covering known as the plasma membrane. So mesosomes are actually formed by plasma membrane folding. So if plasma membrane folding happens, this is called as mesosomes, and this happens in gram-positive bacteria. This helps in DNA replication. DNA replication is a new terminology. I will discuss this in another playlist known as biotechnology. So these are foldings of the plasma membrane. So mesosomes are formed by plasma membrane folding, and they helps in DNA replication and separation into daughter cells. And also it increases the surface area for bacterial respiration. It helps in the cell wall formation also. Chromatophores. These are internal membrane systems which increases surface area for enzyme activity and increases rate of metabolism. So chromatophores are membrane systems, helps in increasing the surface area for enzymatic activity and the rate of metabolism, that is anabolism and catabolism. Cytoplasm. If this is the cell, the jelly-like substance which is present inside the cell is the cytoplasm. But I am mentioning here with yellow color, but it is actually colorless. It is a jelly-like translucent substance, viscous substance found inside the cell envelope. Because outside there is cell envelope, which is again made up of three types, that is glycocalyx, cell wall and plasma membrane. And food is stored in the cell in the form of glycogen and lipids, and it is present in the cytoplasm. Food is stored in the form of glycogen and lipids found in the cytoplasm. Ribosomes found in the cytoplasm of prokaryotic cells is 70S type. Now what is ribosomes? I will discuss this in cell organelle topic. Now ribosomes is cell organelle helps in protein synthesis, and it is found free or attached on single mRNA. So if found free, it is like this, but sometimes it is found attached with single mRNA. mRNA is messenger RNA, and each 70S ribosome is 14 to 15 nanometer by 20 nanometer in size. Ribosomes are formed of two subunits, that is 50S and 30S. So if this is ribosome, the larger unit is the 50S and the smaller unit is the 30S. Okay. Membrane bounded cell organelles are absent in cytoplasm. So in prokaryotic cells, all the organelles which are present inside the cytoplasm or embedded in the cytoplasm are generally membraneless. No membrane is found around the cell organelles. Now nucleoid. If this is a cell, let's take it as a human cell. So human cell is not prokaryotic, it is eukaryotic, that is new type of new cell, advanced cell. So what is nucleoid? Membrane bound true nucleus is absent in prokaryotes. In eukaryotes, there is a proper membrane bounded central part is present, called as true nucleus, and together it forms nucleoid. But in prokaryotic cells, this nucleoid, which is a membrane bound true nucleus, is absent. So what happens? All the genetic material present in the nucleus is dispersed in the cytoplasm itself. They are not confined in a boundary. So nucleoid has a single or circular chromosome with no free ends. It is either single chromosome or circular chromosome but with no free ends. It is formed of double-stranded DNA, not associated with histones proteins. It is present in the cytoplasm with no nucleoplasm, nucleolus and nuclear membrane. If this is the eukaryotic cell, that is human cell, a proper nuclear membrane, nucleolus is present. But you see in this prokaryotic cells, everything is dispersed. Only circular and single chromosomes, that is, these are the genetic material. I know these are new to you. I will explain these things in detail. What is chromosome, what is DNA, but for now it is, you remember that these nuclear materials are found free in the cytoplasm. In some bacteria, extracircular DNA molecules called plasmids are present. So what are plasmids? Plasmids are extracircular DNA. In some bacteria, these are called plasmids. Each plasmid is made up of few thousand genes. So what is the function of plasmid? Some plasmids provides antibiotic resistance. So if plasmids are present in bacteria, some plasmids helps in resistance, gives resistance to antibiotics. Now flagella. So flagella, if this is a bacteria, at the end of the bacteria there is a thread-like structure present, and these thread-like structures are called as flagella. Each flagellum is 4 to 5 micrometer long. It has a basal body that is attached to the plasma membrane of the bacteria, hook and filament. Now what is the function of this flagella? It helps in the movement of the bacteria, that is locomotion. Now depending on the number of flagella, following types of bacteria are found. Atrichous. Atrichous means when the flagellum is absent. Monotrichous means when a single flagella is present. It helps in movement of the bacteria. Amphitrichous, 2 flagella. And cephalotrichous means 2 or more flagella at one end. And lopotrichous, 2 groups of flagella. Okay. This is lopotrichous. And peritrichous, several flagella distributed in the bacterium. This is peritrichous. So flagella function is movement, and it is 4 to 5 micrometer in long, and it is attached to the bacterial plasma membrane. It has a basal body, and it is filamentous in nature. Now Pili or Fimbri. If this is a bacteria, there are hair-like small sized and narrow substance present. These are known as Pili or Fimbri. So in some bacteria there are thread-like or hair-like small sized structures, narrow structures present, known as Fimbri. Pili, long tubular structures. In male there is a special type of pili called as sex pili. Okay, so in male bacteria there is a pili called as sex pili. This is helps for the attachment to female bacterium for sexual reproduction. Pili helps in adhesion.